So this is day 2 of the immunohistochemistry project. So we have our slides that we have stained with the primary antibody, and they've been kept at 4 degrees overnight. We also have all the leftover slides that I showed you from the cryostat. I stayed after we were finished filming and put many slides. So one block of tissue, one axolotl donating its limb to science, can make many slides which can be stained with many different antibodies. For this demonstration, we're just using this one antibody that we have applied, and we'll use one secondary antibody. So after we're done processing the secondary antibody, we'll actually cover slip these. So we'll use that later. So right now what I need to do is make sure that we've got all the liquids together that are in our protocol. So our protocol for today is to wash with PBS, apply the secondary antibody with a counter stain, wash with PBS, add the TrueView, which is an autofluorescence quencher, so that will block any background noise, and then we wash again and then cover slip. So those are the steps I'll be showing you today. I'll show the cover slipping procedure in the next video. What we need to do now is make sure that these get secondary antibody. Now, this negative control is we're gonna put secondary antibody on that as well. Anything we see glowing green on this slide, we know is background. But if the secondary antibody finds the primary antibody on our actual experimental slide, we'll know that it is glowing because of the antibody. So look at the different solutions that we have for our secondary antibody and our counter stain. Notice that the secondary antibody is a yellowish liquid. This greenish yellowish liquid will actually absorb light at a blue wavelength, and it will emit green light, which is what's actually making it a little bit yellowish right now. So you may remember that our liquid from last time that had the primary antibody on it was very pinkish. That pinkish color was the medium that kept the hybridoma cells alive. It was the food. It was not showing us the antibody, but this actually glows. This is actually, the secondary antibody is labeled with an Alexa 488 dye. So this will glow green in our tissue section. This is called our counter stain, WGA, wheat germ agglutinin. And notice how dark, glowy purple that is. This liquid will absorb a yellowish light and give off a reddish light for us on the cell membranes where this wheat germ agglutinin that is conjugated to Alexa 594, wherever that is bound. So we need to mix these two with some PBS. So I've already got a tube of PBS here, and I need, what does this say? 5 microliters of the secondary antibody, which is so small I just lost it in the ice. Now, there's either 10 or 20 microliters in this tube, and I'm only taking 5, so it's quite a shame. Remember, we cannot freeze thaw antibodies. So oftentimes, if you are trying an experiment for the first time, what you may wish to do is just use all the antibody that you have, because I can't put this back in the freezer, it won't work the next time. So there's the 5 I know I should add. What if I were living on the edge and took all the rest of the liquid as well and added that? Now we have more secondary antibody that could bind to our primary antibody, but we're also taking the risk of increasing the background if it binds to the wrong place. So this has to be determined empirically. You have to do this experiment again and again and again to find out what's the optimal level of antibody to use. In the protocol, I said use about 5 microliters, okay? And I just used 10. So the other thing that we need is some of this wheat germ agglutinin, okay? So we're going to take 10 microliters of this dye. So here I've got 10 microliters. There's our purple liquid. And we'll add that. I don't feel any need to add extra of this dye. This dye stains the membranes very darkly. And plus, it was just 10 microliters of each. Instead of 5 and 5, I did 10 and 10. Or 5 and 10, I did 10 and 10. It said 5 microliters of second antibody and 10 microliters of the dye. This, since I added 10 of both, it's a 1 to 100 dilution. 1 mil, I actually can see the purplish liquid here. I think we're going to need to stir that around. So we want to make sure that our secondary antibody and the counterstain aren't all just stuck at the bottom of the tube. So I'm mixing this to make sure that our two fluorescent dyes, which are Alexa 488 bound to the secondary antibody and wheat germ agglutinin bound to Alexa 594, which will give us a red cell membrane and green wherever our primary antibody is. So off screen, we've run PBS through the funnels here. Next step after checking for dried sections is that we run the PBS through. I've shown you how to make the secondary antibody and counterstain solution. So now that the PBS has drained through in our slides, the primary antibody that was not bound has been washed away. The blocking solution that was not bound or was done poking holes in the cells has been washed away by PBS. Now we're going to add our secondary antibody and counterstain in PBS, about 500 microliters to each of our two slides. So what does that look like? Can you see the layer of liquid there in the funnel? It might take a minute for that liquid to funnel through and replace the PBS wash. When that has sat for 1 to 4 hours, we will have given enough time for the secondary antibody to find the primary antibody and bind to it and turn it green. And the wheat germ agglutinin will be bumping into all those cell membranes and turning them all red. So we'll leave this in the dark in a drawer for 1 to 4 hours so that these fluorescent molecules, when they're being hit by the light, they start to glow a little bit. So we want to save all the glowing for under the microscope. So we'll put these in a drawer, and I'll get these back out in a few hours, and we'll look at them.